Hi, I'm Kate Willett from HSI. I'm going to briefly introduce the Animal-Free Safety Assessment Collaboration and the new Masterclass in Animal-Free Safety Assessment of Cosmetics and Cosmetic Ingredients. I'll also provide an outline of the course content and the module release schedule. One of the projects of the AFSA Collaboration Cosmetics work stream has been creation of the Masterclass in Animal-Free Safety Assessment. The masterclass scope includes safety assessment of cosmetics and cosmetic ingredients without new animal data. The course covers all aspects of the process for internal company decisions and regulatory safety assessments, including estimation of consumer exposure, both external and internal, and estimation of different types of biological activity, from acute local effects, like skin and eye irritation, to systemic repeat dose effects. What makes this course unique is that it focuses on understanding the information generated from these tools and how this information is used in decision-making. The masterclass is intended to complement existing courses and webinars that cover how to perform the individual assays. The ultimate purpose of the course is to address the needs of regulatory and regulated communities, CROs and other stakeholders in terms of assessment of cosmetic and cosmetic ingredient safety, and to support regional capacity building in order to achieve long-term acceptance and implementation of animal-free approaches. In the AFSA Masterclass, we have taken the complete animal-free risk assessment process and divided it into 9 modules for content development. The course is based on the ICCR principles and tiered workflow, and is structured around learning objectives. We've included an additional module on the state of the global regulatory environment for cosmetics and chemicals. The AFSA Masterclass covers the 3 tiers of the next-generation risk assessment process in the following way. Module 1 covers problem formulation, including collecting, collating and reviewing existing information and initial MOA hypothesis generation. Module 2 covers consumer exposure, including habits and practices, the data and where to find it, and different types of modeling. Module 3A covers the variety of in silico tools and data and what they can be used for, as well as the thought processes behind them. Module 3B covers the use of in silico tools in identification of similar chemicals and read across. Module 4A covers exposure-based waving, and how it is applied for 3 routes of exposure: oral, dermal and inhalation. Module 4B covers safety assessment of botanicals including history of safe use. Module 5 covers in vitro data, the types of tools used to generate data, and how the bioactivity data can be combined to inform hazard identification, characterization and risk assessment. Module 6 covers internal exposure, including absorption, distribution, metabolism and excretion, the types of models used, and what data and thinking goes into physiologically based kinetic, or PBK, modeling, as well as in vitro to in vivo extrapolation. And finally, Module 7 covers how to put all of this together to achieve a risk determination, including case examples for a wide range of decisions, from acute local, endpoint-specific decisions, such as skin and eye irritation and skin sensitization, to more comprehensive risk assessment for repeat dose, systemic exposures. The masterclass is online and freely available, requiring only registration to keep track of progress. Public rollout of the online modules began in March 2023 with modules 0 to 4A. Modules 4B and 8 are nearly ready, while modules 5, 6 and 7 will be released this autumn. The QR code will lead to registration for the course. You can also visit our website and watch several recorded webinars introducing 4 of the modules: consumer exposure, predictive chemistry, dosimetry, and the global regulatory landscape. You can also find out a bit more about the AFSA collaboration overall. We appreciate the many supporters and active participants who partnered with us in developing this program. And of course the core team of authors that contributed to one or more of the masterclass modules. If you have any questions or would like to learn more about the AFSA Masterclass, or any of the other AFSA or HSI programs, please get in touch. Animal Free Safety Assessment Masterclass. Module 1. Problem Formulation. This module introduces the purpose and process of problem formulation as an important first step in any risk assessment. By determining the overall aim of the risk assessment, problem formulation provides the basis for planning and conducting the assessment so that it achieves the required purpose. By the end of this module, you should be able to 1. Determine the aim of the risk assessment. 2. Describe how to scope exposure, including use scenarios and exposure pathways. 3. Outline the process of searching and evaluating existing information to establish a hypothesis. 4. Explain how uncertainty and bias can be characterized and reduced. Introduction. Problem formulation describes the scope and context of the risk assessment decision to be made, providing the basis for planning and conducting the work needed to complete the assessment, so that it achieves the required purpose. So, why begin with problem formulation? Problem formulation provides a clear aim and structure for the assessment. Problem formulation acts as a reference point to maintain focus throughout the risk assessment. Problem formulation aids in avoiding biases. Problem formulation provides a means for documenting the approach and maintaining transparency. For all of these reasons, problem formulation is an important first step in any risk assessment. To best define the aim of your risk assessment during problem formulation, it is important to consider the decision context under which the assessment is being made. Several context factors will influence the level of information required to come to a decision, the tools that you may use, and the amount of uncertainty that may be acceptable. These considerations include the audience, the type of assessment, the resources available, regulatory requirements, and current best practices. Aim definition example. The scenario: a business partner in the company's research and development team has asked for a consumer safety assessment for a market launch of a new ingredient in a body lotion personal care product at 0.5%. Decision context. Here the safety assessment would require a full risk assessment, including determining the exposure to the ingredient for an appropriate use scenario, and determining a relevant point of departure in order to define a margin of safety, bioactivity or exposure that can be used to conclude safe use, or to identify knowledge gaps. The goal is to determine the margin of exposure for the new product, requiring the determination of both exposure and bioactivity with a reasonable degree of certainty. The market launch is dependent on determination of a high level of safety. The overarching hypothesis for a next-generation risk assessment is whether the chemical is safe for consumer use in a particular cosmetic product for its intended uses. The hypothesis is critical for focusing the assessment and guiding data generation. Hypothesis generation includes 3 key steps: characterize chemical uncertainty, gather biological data, and evaluate the information based on a weight of evidence assessment. Case example: propylparaben. The aim is a safety decision regarding the use of propylparaben, PP, as a preservative at 0.19% in dermally applied cosmetic products, with a focus on reproductive toxicity. Analysis: consumers are dermally exposed to parabens in many cosmetic formulations. After gathering and assessing existing information and conducting an initial in silico characterization, it was determined that parabens have weak estrogenic activity, but there has been no assessment of reproductive toxicity. Therefore, information on the potential to cause reproductive toxicity would be central to the assessment. Hypothesis: based on their highly similar chemical structures, the target chemical PP will have similar bioavailability and bioactivity as the source chemicals methylparaben, MP, ethylparaben, EP, and butylparaben, BP. And the assessment can be performed by applying data from the source substances MP, EP and BP to the target substance, PP. The risk assessment was therefore designed to include investigation of read-across to address repeat-dose systemic reproductive toxicity. Read-across results indicated that PP can be safely used at 0.19%. What comes after problem formulation? As we have just learned, problem formulation describes the context of the decision to be made and provides the basis for planning and performing the work needed for the risk assessment to meet its required purpose. Following the problem formulation, the next step in the cosmetics and consumer products risk assessment process is exposure estimation, as highlighted in yellow in the graphic shown here. If you have any questions or would like to learn more about the AFSA Masterclass, or any of the other AFSA or HSI programs, please get in touch. AFSA Masterclass Module 2, Consumer Exposure. Safety assessments for consumer products, including cosmetics, are led by exposure, which means that assessment of exposure is the first step after problem formulation. And the exposure scenario and predicted exposure will inform the design of the risk assessment. This module describes how consumer estimation is performed, and what considerations go into choosing the types of models used for a given product type and exposure scenario. By the end of this module on problem formulation, you should be able to describe the differences between deterministic and probabilistic modeling. Describe the factors involved in determining consumer exposure to your product. Choose appropriate modeling approaches for different exposure scenarios. Identify appropriate use of deterministic and probabilistic modeling. Assess adequacy of exposure estimate. Compare uncertainty amongst different modeling approaches. Why is exposure assessment necessary? Personal care products, including cosmetics, have direct, often repeated exposure. Because of this close and frequent exposure, it is critical to determine human health safety of cosmetics. The assessment is guided by the intended and possible accidental route of exposure. A tiered approach to exposure estimation begins with looking at the information you have about the chemical, including the composition of your product and how it's going to be used. From this, you can establish the relevant routes of exposure to the product from normal or foreseeable use, and which exposure scenarios are relevant. As a first pass, a deterministic estimation can be made without a lot of data, based on standard assumptions about use and exposure. If no safety concerns are raised, the safety assessment process might stop here, given the worst-case assumptions of the deterministic assessment. However, if a more precise estimation is needed, a higher-tier, probabilistic assessment is more appropriate. If a safety determination cannot be reached, a more precise estimation may be needed, involving a higher-tier, more realistic probabilistic assessment using population data. Routes of exposure. Understanding how consumers are exposed to a product is a key step in the exposure assessment. For cosmetics, there are three major routes of exposure. Skin, or dermal, exposure, including for skin creams, deodorants, soaps and cleansers, shampoos, and shower gels. Inhalation exposure, including for any aerosols or pump sprays. And oral exposure by ingestion, including for toothpaste and lipstick. Habits and practices. To inform exposure estimates, it is important to consider habits and practices, a term that describes how consumers use products. Habits and practices can vary across the globe for the same product type, based, such as the amount of product used, how many times a day, etc. Large databases of this information exist and some are publicly accessible. Deterministic exposure modeling is the most widely used approach for assessing consumer exposure. It generates a point estimate of exposure from simple models using readily available use and habits data. Deterministic modeling is used when data are limited or when a rough estimate is sufficient. It is considered a first-tier assessment. It generally assumes the worst-case scenario and uses conservative data for most parameters, usually the top 90 to 95% of users. Local and systemic exposure. For cosmetics, we consider both the local, external exposure and the systemic internal exposure, which is the amount of the ingredient that is absorbed into the body. Deterministic models for dermal and oral are the most straightforward. Standard values for the parameters used in the daily exposure formulas are provided for several common product types in the SCCS Notes of Guidance, Tables 3A and 3B. The SCCS values, as outlined in this table, can be used to calculate the total or relative exposure to an ingredient in a product. We first determine external, or local exposure, followed by internal, or systemic exposure, which is calculated by applying an absorption factor using simple mathematical equations. The calculations for systemic oral exposure are similar to those for dermal exposure, except an oral ingestion factor is applied instead of a retention factor. Inhalation exposure from cosmetics is generally limited, and usually applies only to products like hairspray, deodorant spray, or powder. To determine whether this route should be considered, you can follow the logic in this decision tree, and ask yourself, what is the likelihood that any of the product will be inhaled? Inhalation exposure can be estimated using mathematical models, either using a straightforward equation particle size, more complex 1 or 2 box models, or if necessary, can be measured directly. Probabilistic modeling. If the results from a deterministic approach are too imprecise and conservative, a more precise exposure estimate could be warranted. Probabilistic modeling accounts for population variability in several habits and practices data, and generates probability distributions as exposure estimates from complex models. Similarities and differences between deterministic and probabilistic modeling are outlined in this table. Aggregate exposure is the total exposure to a substance experienced by a consumer in a given day. It is obtained by adding up, or aggregating, exposures to a particular ingredient in different product categories and through various routes of exposure. The entire consumer exposure module walks you through each of these processes in detail and provides case examples for each step. What comes next? With a well-substantiated and well-documented exposure estimation in hand, you are now ready to move to the next step of the safety assessment. If exposure is very low, you may be able to apply exposure-based weighting, the topic of Module 4A. If not, then you may be able to use in silico tools and read across to inform your assessment, which is the topic of Module 3. If neither of these will satisfy the assessment, you may need to refine your exposure assessment using dosimetry, Module 6, and/or obtain bioactivity information using in vitro assessment, Module 5, before finally evaluating all of the information in a full risk assessment, Module 7. If you have any questions or would like to learn more about the AFSA Masterclass, or any of the other AFSA or HSI programs, please get in touch. Module 3, Predictive Chemistry. To enhance use and confidence in silico tools, it is important to understand the data and processes involved in developing these tools. This module is intended to provide enough information to understand the principles behind the most common types of modeling used in chemical safety assessment. Learning objectives. By the end of this section on in silico tools, you should be able to identify when to use in silico tools, identify the availability of potential tools appropriate for your application, describe different tool types and their applications, evaluate the appropriateness of tool or model choice, identify and evaluate factors contributing to uncertainty. How are in silico tools used in chemical safety assessment? In silico tools refer to any method of prediction carried out using a computer, rather than in a lab. These tools are helpful to predict toxicological activities, such as hazard identification and characterization. to predict biological properties, including informing hypothesis generation and providing information on key events in an adverse outcome pathway, to reduce uncertainty in a weight of evidence, in read-across from well-characterized chemicals, or in risk assessment by supporting or providing missing information. These tools are used during several parts of the risk assessment process. There are many different types of in silico models. For predictive chemistry/toxicology, the most well-known models are QSAR, Quantitative Structure-Activity Relationship Model, and SAR, Structure-Activity Relationship Model. When building a model, the model is only as good as the data it is built from. In general, the more abundant and high quality the data, the better the model. The important aspects of data include relevance, availability, abundance, and quality. The structure-activity relationship, or SAR, is the relationship between the chemical structure of a molecule and its biological activity. The analysis of SAR enables the determination of the chemical groups responsible for evoking a target biological effect in the organism. A SAR model is one that uses a chemical structure to predict its qualitative, biological activity or toxicity. A simple SAR modeling process example. A SAR is identified by looking at several chemical structures as a group and their relative toxicity, and trying to identify the structural features that may be responsible for the toxicity. In the example shown, benzene, toluene, and phenol all have negative skin sensitization data. Paraphenylenediamine has positive skin sensitization data. By assessing each chemical structure, in particular, by comparing benzene to paraphenylenediamine, it becomes clear that the difference between toxicity and non-toxicity is the presence or absence of the amino groups, NH2. Therefore, it is reasonable to presume that the diamino group is causing the toxicity. The module covers different types of SAR models, including decision trees and expert systems, providing several examples. Quantitative structure-activity relationship, or QSAR models differ from SAR models in that they use quantitative measures of chemical structures, defined as molecular descriptors, and correlate one or more of these to a biological activity of interest using a statistical technique. In a QSAR, descriptors and biological activity are used with statistical techniques to generate an algorithm which can predict biological activity from a chemical structure. Hybrid and consensus models. There are strengths and weaknesses to expert rule-based models and statistical models. Occasionally, it can be useful to combine both elements, creating a hybrid model. Module 3A goes on to describe types of QSAR in quite a lot of detail, including linear regression versus classification, development of descriptors, and types and sources of data. Each type of modeling is explained using existing models as examples. Applicability, uncertainty and validation. After selecting and running your chosen in silico model, there are still many considerations that need to be appropriately assessed and documented. One of the first questions to answer before using a particular model is whether the chemical of interest is within the applicability domain of the model. In other words, does the model know enough about chemicals similar to the chemical of interest to give a reliable prediction? If your chemical of interest is similar to chemicals used in the training set, it is likely to be within the applicability domain. If your chemical is not very similar, is far away from the training set chemical space, then the model may consider it as out of domain, and the prediction would not be reliable. Improving regulatory confidence in, and acceptance of, a prediction of toxicity from QSAR requires an assessment of its uncertainty and a determination of whether the uncertainty is acceptable. Understanding and quantification of uncertainty is crucial for risk assessment. Uncertainty assessment identifies limitations in the QSAR prediction and provides transparency to decision makers about what the prediction can, and cannot, tell us. Uncertainty refers to a lack of data or an incomplete understanding of the context of the risk assessment decision. It can be either qualitative or quantitative. It can be reduced or eliminated. Variability is a quantitative description of the range or spread of a set of values. It is often expressed through statistical metrics such as variance or standard deviation. It refers to inherent heterogeneity of diversity of data in an assessment. It cannot be reduced, but it can be better characterized. Uncertainty and variability arise from the data used in the model, the modeling approach, and the modeling application. The module goes into more detail about the sources of uncertainty and variability, including how to estimate and communicate them. Validation is the most crucial concept for the development and application of in silico models, as it allows you to evaluate the predictivity and reliability of the model's predictions. There are two types of validation: external and internal. The module describes well-developed validation principles and discusses different types of internal and external validation. So, what comes next? One important application of in silico tools is for identification and characterization of chemical groups or families for the purposes of read-across. Read-across is an alternative approach that is used to fill a data gap for a substance, the target, for a specific endpoint, by using the data from another structurally or mechanistically similar substance, the source. The read-across assessment process is the topic of Module 3B. If you have any questions or would like to learn more about the AFSA Masterclass, or any of the other AFSA or HSI programs, please get in touch.